Okay, let's get started. Welcome everyone to another episode of Hanover Science Seminar Series. Today, we have Chip Frazier from Virginia Tech, who is a plant polymer scientist. He's the Thomas Brooks Professor of Sustainable Biomaterials and the Director of the Wood-Based Composite Center. So let's give a round of applause and welcome him. And then we are very grateful you accepted our invitation and for this talk. So I will hand it over to you and please go ahead.
Thank you.
But
Thank
yeah,
you so much.
that's what I just emailed them back.
Thank you for the introduction and thank you for the invitation to speak at this great university. I've been here 30 years ago, and it's wonderful to come back for a visit. It's even better to be invited to participate in the James Hanover Symposium series because I have a connection with Jim Hanover. I went to graduate school with his son, Carl Hanover, at the University of Washington in Seattle. And we were roommates. And so it's just special to be participating. And because of that, I wanted to just kind of highlight some of Jim Hanover's research because it's interesting to me. And I was surprised to learn about his early work looking at epicuticular waxes. He was studying that. He was demonstrating why blue spruce has the blue color because of the reflectance. And it's a very fascinating topic. I love these waxes. The wax in this particular case is non-icosanol, which is a chiral molecule. It makes chiral crystals. And so this work that Jim did is still pertinent today because folks are interested in non-icosanol tubules for nanomaterials. Also, of course, Jim Hanover did quite a lot of work with terpene analysis for forest genetics. And that's where I first learned about his research. Because at the University of Washington, I was studying terpenes in Western red cedar. And so that's when I first learned of Jim Hanover's work, because he had done quite a lot in terpenes. And so, again, it's just a pleasure to be invited here to participate and to be associated with the James Hanover Symposium. I'm just very appreciative of that. Thank you again. So let's talk about new materials through the enigma that is lignin. Whenever I talk about lignin or whenever I think about lignin, I actually think of old materials. I think of evolution. And so let me take you back. So we believe that photosynthesis started about 3.5 billion years ago, and that the land plants showed up much, much later, billions of years later. And of particular interest to me is the Carboniferous era, which was basically created because of lignin. So here's the beta aryl ether structure of lignin. I will feature this prominently. And lignin evolved, as we know, to protect cellulose and to bind it together. And it did a great job. In fact, it did such a wonderful job that during the Carboniferous era, for about 60 to 80 million years, when trees and plants died, they simply died and fell over. They were inert. There was nothing that could biodegrade them. They became covered with sediments, and then through geologic process, they were transformed into coal. So that's the first material science concept I like to discuss, and that established the carbon cycle on Earth. So that's a big deal. And when I think about evolution, I often analogize it to the Borg because I'm a big Star Trek fan. And when I consider cellulose, that's just a single mode or a single frequency shield. There's not much protection in that. Fungi and bacteria probably only need about 5,000 years to figure that out. So that's pretty easy. But a multi-mode, a multi-frequency response, that's what lignin created. And that was far more effective in terms of evolution. But nevertheless, resistance was futile. Ultimately, fungi and bacteria did figure out how to get around the protective structure of lignin, but it's still an issue. And it's a poor analogy, really, because the evolution is an ecology of interacting organisms. It's not a conscious competition. And so circularity was achieved in a relatively brief time. 60 to 80 million years is relatively brief. But what I find remarkable about this, even with this cloak of variability, which is still very important for lignin, it's remarkable that so much of the lignin structure is dominated by the beta aryl ether. So on the one hand, the organism decided, let's have a variable polymer. On the other hand, there's anywhere from about 40 to 60% of the lignin linkages are beta aryl ethers. So it's variable on the one hand, but not so variable on the other hand, and that's good because nature left beta aryl ether, enough of it that fungi and microorganisms were able to get around it. And so otherwise, if lignin had been too efficient, then a carbon cycle would never have emerged. Now, one of the things that is so promising about lignin chemistry today is that the beta aryl ether linkage, the chemistry of this linkage is very well understood. And I think there's a new era in lignin activity, which basically suggests to me that there's so much beta O4 linkage in lignin that a practical and simple way to deal with native lignin. Now, I must point out native lignin, not technical lignin such as Kraft. A valid way to deal with native lignin is just to treat them as a beta aryl ether homopolymer and just focus on that functional group alone. Of course, it won't be completely efficient, but it's highly efficient. And as you know, lignins differ. Angiosperms exhibit a higher beta aryl ether level and fewer carbon-carbon linkages. So the carbon-carbon linkages are red here. And in gymnosperms, we have a lot more of that. And that's a great practical significance and frustration for microorganisms and humans. So the higher carbon-carbon bond content of gymnosperms is a significant issue for us as well in the isolation of lignin. But normally when we think about lignin, or when I think about lignin, it's often in the context of CO2 levels, carbon dioxide levels. But lignin is also associated with oxygen levels because it's an oxidative, it's an oxygen-dependent reaction for the polymerization of lignin. And for billions of years, there was simply no oxygen. And then when photosynthesis emerged, oxygen started being produced, and it took about 2.5 billion years for that to titrate the iron and other compounds on Earth. And it wasn't until about 600 million years ago that we started to get atmospheric oxygen accumulating to the level that allowed life to proliferate the way it did. And when it did so, well, in these years before, thankfully, the phenylpropanoid metabolism was developing, which we needed. And also these peroxidases, lactases and peroxidases evolved and gave us the opportunity, or created the opportunity, to polymerize lignin. And so when we think about this shield of variability, it's because of aromaticity and resonance. So in evolution, lignin was an innovation, and behind that innovation was the aromatic chemical nature of lignin. And that resonance was possible. For instance, if we simply look at the three most prominent monolignals, I'm showing them now in their oxidized state. These are radicals. Everywhere you see red is a reactive site. And then, of course, as many of you know, as the structure changes over to the S unit, you have fewer reactive sites. And so angiosperm lignins tend to be featured here. And so we tend to have mostly reaction of the beta position with the oxygen, giving us the beta O4 or the beta-beta reaction. But as we increase the number, or the monolignals, we start to get a greater level of carbon-carbon cross-linking, as you know. But in terms of the variable shield, what was so radical about this was the resonance stabilized radicals, and that after they were formed, they coupled and polymerized in a non-enzymatic fashion to produce something that's somewhat random. And so that was a huge innovation. Lignin, the fact that it's aromatic, that takes us into an entirely new realm of chemical reactivity. And I don't know if you knew it or not, but we've had a love-hate relationship with formaldehyde in the wood products industry for a long time. And it dates back to the 1930s. And back in the 1930s, it was a love relationship. Because back in the 1930s, folks were trying to determine what the actual structure of lignin was. They had no idea. Well, they had some idea. And at the time, they knew that when you heated wood, it generates lignin. It generates formaldehyde. And it generates a lot of formaldehyde. And back in the 1930s, it was well established that the formaldehyde came from lignin. And so back in the 30s, the explanation of formaldehyde generation was a key in order to get the structure, the conceptual structure of lignin correct. And when they did, that was basically when they established the phenylpropanoid structure of the beta-O4 unit. So formaldehyde has been important in the wood products industry forever, but back in this age, it was actually loved because it contributed to our understanding of lignin. And sadly, that understanding was lost very dramatically. We lost this understanding, or at least it wasn't retained by some groups. And now the love-hate relationship is, today's discontent. That's how we typically view formaldehyde in a negative light. And that's because of regulations to limit formaldehyde emissions from non-structural composites like these that we have in our homes. Back in 2010, that was when President Obama signed legislation to create what effectively are the current formaldehyde regulations. Those regulations that started in 2010 actually became law in 2017, but it all really happened in 2010. And so at that time, the regulations to minimize formaldehyde emission from non-structural composites, it became very, very rigorous. In fact, so rigorous that the natural formaldehyde generated by wood, when it's heated, started to complicate the compliance with the emissions regulations. And so this, in 2010, this was when the members of the Wood-Based Composite Centers, an industrial research consortium that we have, they asked my group to start studying wood to determine how it made formaldehyde. And at the time, I had no idea that it was a lignin reaction. And like I mentioned, it had been, to a large degree, forgotten. So we started studying this. And then we discovered that, yes, indeed, lignin is the source of the formaldehyde. And I have to say, as I was talking with the graduate students earlier today, that this was a transformation of my career. When the industry asked us to start studying formaldehyde generation, I wasn't exactly excited about it. It's been a topic in wood composites forever. I had little interest in it, but they were my sponsors. And so I responded. And I'm so thankful that they asked me to do this because it changed my career. I was a resin chemist, and then I became a lignin chemist. And now my entire effort is focused almost exclusively to lignin chemistry. So big changes can occur in life. And that's what happened. So back about it took about 7 years for us to start publishing after that change. And I had some students that we were just studying the formaldehyde content in wood as it exists naturally. And we were studying how formaldehyde is generated when wood is heated. And one of the things that we discovered that wasn't fully appreciated is that there's a very strong species effect on the generation of formaldehyde. So looking at this data here, the blue data shows when the dried wood was heated and it generates formaldehyde. And so what I want to point out is that radiata pine, plantation-grown radiata pine, has unusually high formaldehyde emission levels. And we really do not understand that. And so when products come from Chile, for example, from Chile, the resins that they use to make products from that type of radiata pine has to be adjusted to get the formaldehyde compliance. And so it's a big deal that folks have been dealing with. And we studied that for a while. And then as we continued, we started to look into the specific reaction that generates formaldehyde. So here again is the beta-aryl ether. And the first step in the reaction is the protonation of this benzyl alcohol to lose water, and you form a carbocation. And at this stage, this reaction can go two different ways. It can go this way, C3, or this way, C2. And over here is where formaldehyde is generated. And so that's the reaction that we were interested in. What we discovered in this work, or what was known before we started doing this, was that with model compounds, small, simple compounds, that you could selectively choose one pathway over the other exclusively with model compounds. And so we wanted to know if these reactions were occurring in solid wood, and yes, they are. And what we discovered is that using different catalysts, we are able to promote one pathway more than the other pathway, as the theory predicted. It wasn't exclusive catalytic selectivity, but it was mostly one way or mostly the other. So we were very excited about that to learn that, yeah, okay, we think that we can control chemistry in solid wood with catalytic selectivity. That would be very interesting. And let me point out one important point is that in this process, in native lignin, this is called lignin acidolysis, this reaction. And when it happens, if it goes this way, the products cross-link very rapidly. And that's why I call lignin the amazing self-healing polymer. Lignin, the ether linkages break easily, and they reform carbon-carbon linkages that are much stronger. That is an incredible capacity for any polymer, and that's what lignin does. And so if it goes the C2 pathway with formaldehyde, the cross-linking is very rapid. If it goes the C3 pathway without formaldehyde generation, the cross-linking is not so rapid. But this is a huge deal. So think back to our story of evolution. We were talking about variability in the structure. That was important. Well, here's yet another level of variability, but it's after the organism is dead. That is, these linkages break readily, and then they reform carbon-carbon linkages. So not only is lignin variable in its evolution, but after it achieved that status, it had another trick in its bag where it could vary the structure even further. Oh, something went wrong here. Sorry, there's an error in this slide here. That's Gui Gui. She worked on this project. Subsequently, Mohammed Tasuji, who's one of my grad students, one of my most recent grad students, he was working with Arauco in Eugene, Oregon. They have a medium density fiberboard plant there. And they actually gave Mohammed full reign. For 2 days, they let him operate that mill for his research purposes. I'm sorry, my slide is all messed up here, but what Mohammed demonstrated in his work is that in the thermomechanical refining of fiber, at different energy levels, different quantities of formaldehyde was generated because lignin is incredibly reactive. And so in the thermomechanical process, formaldehyde would be generated. And Mohammed demonstrated over here, showing a higher formaldehyde levels. And this is Douglas fir and the formaldehyde content without refining. And this is refining at 2 different energy levels. And so what we found was that it was sensitive to the formaldehyde emission, or that the energy used in refining was sensitive to formaldehyde. And so we believe that it's possible to use formaldehyde, online formaldehyde detection as a process control, and that may be true in the future. We'll see about that. More recently, my slides are really messed up. I'm not so sure why, but more recently we had some work. We're looking at trying to promote in situ lignin cross-linking, and we did that. So we wanted to know again if our hypothesis was that we should be able to promote the lignin reaction in solid wood. And I'm so sorry that this slide is messed up. I'm not able to show you that effort, but the bottom line was from that work was that yes, we demonstrated that the reactions that we expect to occur in wood, they do occur in wood. And so we believe that in the future, we're going to develop a new method to react with that. So in the lessons that we've learned in recent years is that native lignin is very reactive. It favors cross-linking. And so where we're going with this is to go with that and to work with lignin. So even at temperatures as low as 40 degrees centigrade, that's where formaldehyde generation starts. And so that's where the lignin reaction starts. So we're very interested now, what we're in the process of now is synthesizing lignin crosslinkers. We are using C10 flexible segments and then just terminating them with different aromatic nuclei to participate in the same types of reactions as that lignin participates in. And so this is where we're headed. And so the native lignin, very reactive, even drying at 100 degrees centigrade, reduces or produces formaldehyde. And so that reduces the functionality of the lignin, and that's very important for us. So the work that where we're headed is to develop an in situ, a lignin-specific reaction in solid wood. So that would, we want to actually increase the cross-linking and alter the lignin structure and perhaps introduce new functionality. Now, this approach that we're taking is a kind of a twist on an old term, lignin first. Lignin first. So this is our approach here for lignin first. Oh, I'm so sorry. Oh, my slides seem to be messed up. But for a lignin first approach, the question is, what do we do? Do we take the lignin out first, or this is supposed to be cellulose? Which goes first? And the lignin first strategy says, preserve the natural structure of the beta aryl ether. That's the lignin first strategy, and that's what we're taking. But I want to comment on, I'm going to switch gears now and talk about a new discovery that was made recently. So when we talk about the lignocellulose biorefinery, we want to fractionate. That's what the biorefinery is all about, fractionating lignocellulose, but we want to preserve the lignin. So the lignin first strategy is to remove lignin while preserving its native structure. That's the big deal. And when we talk about the great enigma of lignin, when I talk about new materials from the enigma that is lignin, it's this self-healing reaction that is the problem. So here's the basic reaction that we've been talking about. The beta aryl ether, it undergoes lignin acidolysis. It can go this way for rapid cross-linking, this way for slow cross-linking. Working with lignin, one would preserve the beta aryl ether and prevent the self-healing cross-linking that I mentioned. But the question is it economically feasible? And we'll discuss that. Working against lignin is really what we've been doing for over 100 years. We know how to remove lignin, but we don't know how to remove it yet by retaining its structure. And so when we talk about the biorefinery, a kraft pulp mill can't be considered a biorefinery because a biorefinery will produce high value from all streams. That's the objective. And currently with kraft lignin, it doesn't have a high enough value. However, when you look at the work that Dr. Najad has conducted, maybe that will change in the near future. But as of now, we're interested in trying to preserve that natural beta aryl ether structure. Oh, I'm so sorry that all of my slides are messed up. So I'm just gonna wing it now. So what happened is, in terms of a lignin first strategy, there was a report in the literature about a year ago. It was made by Li Shuai, the author of this paper that you cannot see. Shuai reported on the new method of isolating lignin using formaldehyde. And the lignin, turns out, is a very good thermosetting resin. When you bond it with wood, it actually resists boiling water. And so this was a big discovery. This came to my attention. This article was published in Nature, and Nature asked me to review the article. So I reviewed the article, and they asked me to write a review of the article, which I did. And it was very fascinating because this work started in a prior year by Li Shuai. I'm not sure if his name is up here, Li Shuai. So it was Li Shuai and Jeremy Ludebacher, right here, who published this article several years earlier when they demonstrated a way to isolate lignin using formaldehyde. And the formaldehyde, what's so amazing is that it reacts with the aliphatic hydroxyl groups in lignin, and it forms a dioxane ring structure, which is lovely. I wish I could show it to you. But instead, all I can show you is this image of myself. There's Lee and me. I had to put the highlights here because you couldn't see me otherwise, right? So we were at the Gordon conference several years ago. So at the time, Virginia Tech had hired Lee Schwei. We hired him, and it was after his publication of this new method to isolate lignin using formaldehyde. And we were working together, but unfortunately, we were unable to retain Lee. He returned to China and continued working. And then it was some number of years later that he published this paper. And this is how the whole story started. So with Ludebacher, back in 2016, they developed a method using formaldehyde as a way to isolate lignin. So it was several years later that they demonstrated that this so-called formaldehyde-protected lignin is a very good structural thermosetting resin, hydrolytically stable, which is very important. That means it has a very low formaldehyde emission. And perhaps for the first time ever, an isolated lignin actually has high value. That's the real excitement about this development. So here is the basic reaction. So there's a large excess of formaldehyde, and here is the beta aryl ether structure. And look at these two lovely hydroxyl groups that will react with formaldehyde, produce this 1,3-dioxane structure, beautiful structure, very stable under alkaline conditions, but under acid conditions, very, very reactive. And Mojgan asked, Dr. Najad asked a great question earlier in the day about this reaction. So the reaction with formaldehyde is a protection. So having this acetal structure here protects the beta-O4 linkage from the self-healing reaction. So if the beta-O4 linkage is protected, how do we remove the lignin? The lignin has to be removed. So the chains must be cleaved. So it's a very fine equilibrium with a large excess of formaldehyde. Most of the time we're over here, but it's an equilibrium. So sometimes, there's always a very small quantity of the native structure. And under acid conditions, it will cleave and undergo the self-healing reaction. Sometimes it's not successful in self-healing, and we can remove those chains. And so it's a very subtle control of equilibrium. And in the interim, so this is a fascinating polymer. It looks like it has higher value than lignins normally do. So Lee Schwei left Ludebacher's lab, but Ludebacher continued to work on this. And what I would show you if this slide was working was that they developed ways to isolate the lignin using different aldehydes. So this lignin isolation is an aldehyde-assisted isolation. Formaldehyde, it turns out, seems to be the best, but you can use other aldehydes shown here. So formaldehyde-protected lignin, but very important, developed by the Ludebacher lab, is glyoxylate-protected lignin. That's very important because formaldehyde-protected lignin, this lignin is water insoluble. It's soluble in organic compounds. But the glyoxylate-protected lignin with this functionality can be made more water soluble or more water dispersible, and that's very interesting. Also, about each of these, so, we have many different aldehydes that we could use. And so, for instance, you could use furfural during the isolation. You could use glyoxylate during the isolation or terephthalide. But to use those aldehydes in the lignin isolation seems to be inefficient. So what it looks like now is that it seems that the economics of lignin isolation may be much more favorable with formaldehyde because formaldehyde is a chemical of scale. It's a huge scale and it's very inexpensive. And these compounds are much less expensive. Much more expensive, pardon. So in order to generate compounds like this, it looks like it might be necessary first to produce the formaldehyde-protected lignin on a very large scale, and then to conduct an acetal transfer reaction to get to some of these more interesting structures. So, these are more interesting structures. But for me, I'm fascinated by this structure. This structure is sufficiently interesting Because again, I'd like to emphasize, for the first time ever, a lignin isolation product is right out of the box a structural thermosetting resin. Not just for wood, it works for wood, but if it's a thermosetting resin, it could be a matrix resin for numerous applications. So this is truly a big development. These slides are killing me. And so the implications for this are the new biorefinery, the future biorefinery. And so we can envision a process where we bring in lignocellulose to manufacture numerous things. So we'd bring in wood or some lignocellulose source to isolate the lignin, and that lignin then could be pumped over to an adjacent mill that's manufacturing a wood-based composite. In the isolation of the lignin, the cellulose, of course, has to retain its value. And that's the big question in the isolation process is the cellulose and the hemicellulose isolated? Are they pulp and paper quality cellulose products? That's going to have to be answered. Remember, for a biorefinery to be real, each fraction has to have high value. And so for formaldehyde-protected lignin to be useful, the cellulose has to be just as good or better and less expensive or the same cost as before. So big implications for the biorefinery. So summarizing and apologizing for my poor slides, native lignin, very reactive, easily the most reactive plant polymer in lignocellulose. Lignin first strategies preserve the beta aryl ether. And this is where my focus is now, on lignin first strategies. I am interested in preserving the beta aryl ether and then using that. So right now we're working on lignin specific reactions in solid wood that we're interested in. And we're also interested in formaldehyde assisted lignin isolation to use FPL as a new adhesive. So formaldehyde protected lignin is perhaps a major breakthrough towards the realization of the biorefinery, possibly. And if so, the irony will be off the charts. I'm not sure if the students appreciate this fact, but formaldehyde emissions from wood products have been an issue since the 1980s. And formaldehyde has been the ugly child in the wood products industry. But formaldehyde adhesives have been highly effective. And so there's just this give and take. Some of us would like to get rid of formaldehyde adhesives, but there's been no alternative that's economically feasible and as effective. If this new breakthrough has merit, well, our relationship with formaldehyde is going to change. There's going to be much more formaldehyde. And perhaps a new level, perhaps it will be less hateful, because if formaldehyde in the end allows us to achieve the biorefinery that we desperately need, yes, it's going to change the perspective on formaldehyde, I believe. And I'd like to thank you for your attention and acknowledge my funding sources, the National Science Foundation. This should be USDA over here. And then the biggest one is the Wood-Based Composites Center, an industrial research consortium that's existed since 1999 and has changed my career. That interaction with the industry has been a huge boon for me and my students. And I thank you for your attention, and I hope that there are many questions.
We always start with the students.
All right, ask questions for the one.
Don't be shy. I'm gonna give each one.
Yeah, well, if you have any questions.
Okay, I gotta start.
Sure.
Make sure that they feel more comfortable as questions. You mentioned that you're interested in using formaldehyde to extract lignin. One of the main problems that we have with formaldehyde is free formaldehyde emission. If you measure that, like after extracting lignin, how much free formaldehyde is in the lignin, or through your process you are removing all the free formaldehyde?
There must be free formaldehyde, and so that formaldehyde has to be stripped. Yes, but the good news is that it can be stripped. So it's not even in methylol form. It's actually free formaldehyde. So yes, in this process to produce this lignin, there's such an excess of formaldehyde that has to be stripped out. The formaldehyde that's incorporated chemically is incorporated in carbon-carbon bonds. And so it's hydrolytically stable. And so it's not going to be an issue with formaldehyde emissions. Yes.
If you're not asking.
Okay.
Yeah.
Oh, sorry. I was wondering if, I think it was your second, in your summary slide, your second bullet point about that. Yeah, so you were talking about in situ, in the wood reactions to introduce novel wood.
Yes.
Curious if you could expand on.
Yes.
that one. What kind of new properties
Oh.
are you looking to achieve there
Oh.
with that process?
Oh, a great question. Well, the holy grail for wood material science is hydrophobicity and fire resistance.
Okay.
But I'm interested in some other things like magnetism also. But the answer for the industry would be to make wood more hydrophobic and more fire resistant. And so this is our plan. Not that it'll work, but we're going to find out, I hope. We're synthesizing compounds like this to react with lignin and cross-link it. And cross-linking is my immediate interest, but it doesn't have to be for cross-linking. Basically, it's just a lignin-specific reaction that we hope that we could introduce some other functionality. And I'm interested in magnetite nanoparticles. I don't know why. I had a great collaboration with Judy Riffle, professor in chemistry at Virginia Tech, and she was working on magnetite nanoparticles. And I don't know, ever since then, I've just been interested in it. So.
If you can make them to enough that they can absorb other proteins, I think you're going to have much better achievement.
Yeah.
Get a lot better.
Oh, yeah. Yeah.
And if you're going to use this in a process, say, by adding this, we can make it more conductive down.
Yeah, well, thank you. That's the idea here, really. This idea, I don't know if it has any merit. I mean, at present, I'm just interested in it as a fundamental exercise. But if it does have merit, where I envision it occurring perhaps in oriented strand board manufacture right after flaking, there would have to be some type of a treatment to get this into the cell wall. Because we really would like to get this into the cell wall before drying occurs, because there's enough energy in drying to kick this reaction forward. And then more reaction would occur in the hot press. So this lignin first strategy is one that says, first derivatize the lignin and prepare it for reaction in a subsequent composite material. Yeah.
Great, thanks for coming. Go back to the C2, C3 path.
Yes.
I want to see what are you calling C2 and C3? How is it formaldehyde forming?
Let me go back to the previous, or do I have it? No, I'm sorry. So the C2 pathway is the one that produces formaldehyde, and it's called the C2 pathway because in the aliphatic chain, there are two aliphatic carbons remaining. Yeah, and then the C3 is because you retain all of the carbons. That's the nomenclature. Correct. So yes, yes. Yes. So now we're talking about the addition of synthetic formaldehyde to react with the lignin. Yes. Yes, you do. Thank you for that question. That's a great question. Not nearly enough. Because actually, we have to preserve that formaldehyde. So that carbon there, we're going to lose it. And its reaction with formaldehyde is gonna tie it up. I have the wrong structure here, but yeah, you need to preserve all of the lignin formaldehyde. Otherwise, if the lignin formaldehyde comes out, then the beta O4 linkage is destroyed, and you've lost the value. So the real value, my point is, the real value is the original beta aryl ether.
So you want to see the pathway to occur.
Another great question.
And then you're
The C3 pathway is preferred just because of the protection. So once the lignin is protected in that acetal, I may have this, I'm not sure. I've lost track. My poor slides. Yeah, I'm even missing slides. I don't know what happened. I even lost track of what the question was, so I'm so discombobulated. Yes. Yeah. So yeah, so when it's in the acetal form, when these two hydroxyls are reacted, when it opens up, that product is probably going to promote the C3 pathway. Promoting the C2 pathway is of interest simply because this compound homovanillin is so reactive. And of course, you have formaldehyde, which is so reactive. And so there's some theoretical interest in promoting the C2 pathway, but I'm not sure it's possible.
But even if you did, you wouldn't be able to balance the formaldehyde you need to do what you want to do with the C3 pathway, right?
Well, again,
You're gonna have to come without that.
yes,
There's
yes,
implications in regards to chemistry.
yes,
Somebody's got to handle that.
right. No, that's true. So yeah, I mean, I am involved with the industry on the commercialization, and so the formaldehyde issue has been a big one. I can't say much more than that, but the circumstances in Europe and the United States are different currently.
Question in a moment, but you mentioned that you're really targeting to show that structure with large alpha chaining with a C10, if I counted correctly.
Yes, correct. Yes, decade.
So the gold standard these days isn't just hydrophobicity, it's resistance to water and oil. And so any thoughts is what you might put in there. This is not a big challenge, but any thoughts you might put in there to resist water and oil, so they both be.
Well, I guess I'll push back a little bit. That's the gold standard in some industries. I'm aware of that. I'm aware of that. So for us to get wood to be hydrophobic in an economically feasible scenario, that would be great. And, you know, is acetylated wood that? Perhaps. Perhaps. I'm not sure. Yeah, but yeah. Yes. Right. Right. Yeah. Yeah, I'm asking my packaging colleagues about the PFAS issue as well. So yeah. Great question. Yeah. 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 Yeah, fluorinated cellulose, it didn't go over so well. Teflose, I was going to call it. Yes, sir.
I guess it might be a little more markets based, but I thought your comment in your slide was interesting about you maybe have achieved this high value lignin. Now the question is, does that cellulose and cellulose have that high value? And I'm wondering about how market trends might affect that if we're going away from less nice office paper and more
Yes.
Amazon cardboard boxes as the driver.
Yes.
Does that change that? Do you have any thoughts on?
Only I think where you're headed. I mean, it's just that whatever the properties of that cellulose are, it's got to be good, right? Or the whole thing is useless. So the alternative, I can tell you that the alternative aldehydes. Oh, wow. The alternative aldehydes. Let me see. They're much more expensive. They have greater utility in some cases, but their ability to pull the lignin out is so much less. So we can use these pure aldehydes to isolate the lignin, but the difference in size has a big impact on the yield, and so that's going to be a part of the process. Oh, yes. And as I understand it, I've remembered now, in the using formaldehyde, the reactivity of formaldehyde apparently ties up the surface of the cellulose to a substantial degree. And so strong acid has to be used to get that formaldehyde off. And so the question would be, okay, what's that cellulose? Then what is it best suited for? Is it best suited for energy or paper or regeneration? I mean, that's the kind of thing that has to be worked out to determine what that does to the cellulose, because these other aldehydes have very little impact on the quality of the cellulose. They're not as reactive, but then they give a poor yield. So it's always this balance, right? We're trying to find the sweet spot.
Thanks.
On that note, Chip, does acid peel the formaldehyde off does it break the dioxane and bring it back up?
Yes.
So you could then react it subsequently with terephthalaldehyde.
Correct.
And go into hydrogen or whatever else.
Correct.
That's reactive on the other end.
Correct. Yes. So an acetal. So instead of an ester transfer, acetal transfer reaction. Yeah. I'm very anxious to try that because introducing just a little bit of glyoxylate, I think, is of interest to me. Because right now, if this formaldehyde protected lignin is actually a structural thermosetting resin to make plywood, that's great. But to make a bigger difference in society, it needs to be a non-structural resin. It needs to replace urea formaldehyde. And to replace urea formaldehyde, it must have tack. It must be sticky. When you resonate the wood particles, if the wood particles don't adhere like a snowball, then you cannot make particle board or non-structural composites. And formaldehyde protected lignin is organic soluble. It will have no tack. You could introduce tack by giving it a little bit of water solubility, water dispersibility. That will give it tack. And I think introducing a little bit of that with a lot of this might be a way to get to a non-structural resin for particle board. But it's all talk. And I've only got 4 years left. I retire in 4 years.
One of your slides about the role of stages, how different stages are getting that.
Yes.
Just curious if because with all of this climate change impacts, like the drought and all of the conditions that have you had the chance to look at the impact affecting lignin production or even just isolation? I even don't know what I'm asking. I don't really understand impact.
Yes. It's a great question. Because when we try to envision the biorefinery, the vision is that it's located next to the resource and next to the infrastructure for using it and for transporting it. And so when we talk about the yield of the formaldehyde protected lignin, the yield is low in softwoods, from softwoods. The yield is high from hardwoods. But the market for wood-based composites is mostly softwoods. And so there's kind of a problem there already. I mentioned cannabis. So if cannabis was a good feedstock, then how does the cannabis industry relate to the rest of the forest products industry logistically? You know, I think it's probably pretty good. I think cannabis industry is growing from the Southeast all the way across. And so logistically, it may be good for cannabis because it's an angiosperm. So I don't have an answer, just like your question, but it's an important question because the feedstock, if we could use softwoods and gymnosperms, that would be great because I think we're processing more. If we can use cannabis and if it's economically feasible, then that could be very big. But it's all talk at this point. Thank you for that question. Enoch. Yeah, thank you for that question. So the yield of the lignin is very near the theoretical yield of let's say vanillin or beta O4 linkage. So if we assume 25% lignin content in an angiosperm, the lignin yield is over 90% on a theoretical basis. So it's very high. With gymnosperms or with softwoods, that yield plummets down to about around 50% of the theoretical. So it's a huge reduction. And I forgot other aspects of your question. The curing, yes, that's very important. So here is the formaldehyde protected lignin and the curing mechanism, it's acid catalyzed. So I envision this as the first step in the reaction. So this dioxane unit has to open up. It has to reveal this benzyl hydroxyl group. So then it can react in the normal lignin acidolysis pathway. And so once this pops open, and Chris, I think that's why I think it's a C3 pathway because the gamma hydroxyl is still protected. So I think it'll open up in this fashion, as opposed to opening up here, I think it opens up here probably because of the stability here. And so I think it's going to favor that. But the reaction is then, once this opens, then it's the lignin acidolysis cross-linking reaction that I mentioned. And also, actually, if we consider the acetal transfer reaction, so if we have this acetal and we wanted to react it with another aldehyde, as I mentioned, we first have to, the acetal has to break open so we can react it with another aldehyde. But at that moment, of course, lignin acidolysis could occur. So there's probably little doubt that by starting with formaldehyde and then switching to another aldehyde, the yield is probably going to be compromised because of this. You're going to reveal the native structure to some degree in an equilibrium. And if you do, it'll cross-link because lignin is incredibly reactive. Perhaps that is the best message from today, the one I'd like to emphasize. Native lignin is incredibly reactive, more so than I think folks have appreciated. So certainly more than I've appreciated. I'm sorry, could you repeat that? Oh, no. No. See, the requirement here is native lignin. So using this extraction on craft lignin, in craft lignin, the native structure has been destroyed. So we need to have this beta O4 structure. So that's why whenever I hear about a new lignin, my question is how much beta aryl ether, how much beta O4 structure does it have in it? And most technical lignins have very, very little. The formaldehyde protected lignin has almost all of the original beta O4 structure, but in the acetal dioxane structure. But it's still very reactive. Still very reactive. So the answer is this will only work with native lignins, not with any type of technical lignin. So this is a new way to produce what will be a new technical lignin. And hopefully it'll be successful. And how long will it take? That's a good question. So there's scientific merit here. What do we think? How long will this take? If this actually can work, how long will it take before it's even pilot scale? That could happen in a couple of years. How long would it take for production? That might be 10 years. That's the thing. It's so worrisome. You know, there's a great climate change urgency, you know, of course. And so it takes a lot of time to do this stuff. We'll see.
One question? We are out of time. Yeah. So I would like to thank again for Dr. Frazier to come here.
Thank you.